So in this video we're going to learn about the genetics, particularly Mendel's law of inheritance. So first of all, what is genetics? It is the branch of biology which deals with the study of inheritance of the genetic characters. This Mendel, Sir Gregor Mendel, he was known as the father of genetics. Now why he was known as the father of genetics we'll be learning in this chapter. So what he did was, with the help of his experiments on a garden pea, he was able to formulate laws which explained the process of inheritance of characters, and due to this contribution he was known as the father of genetics. So he was able to explain by the help of his laws that how the characters are inherited from the parents to their offsprings. So for this experiment he selected garden pea. Now why did he select the garden pea? We'll learn first of all that. So the reasons for the selection of garden pea. Now this question is asked many times, and I'll tell you one information from this chapter. In the last 23 years, now see, there are 5 sets of questions for the 5 development reasons A, B, C, D and E. So basically in the last 23 years, out of these 5 sets, the questions from this chapter in the 3 marks, it is asked all 23 times. It means in the past 23 years this question is asked 23 times, at least in one out of the 5 sets. So this is one of the major chapters, and this is about 3 marks in the 8 marks or 7.5 marks question. Usually it is asked in 8 marks. It is asked 5 times in the past 23 years, but in 3 marks this question is usually asked many times. Any question can be asked, like the reasons of the selection of the garden pea. Then we'll further read about the dihybrid cross. It can also be asked. So first of all, let's see why the garden pea was selected by Mendel. So the first reason was that the garden pea, it possesses many unit characters. Now what it means? It means that the garden pea, it has many characters which can be studied, like its seed color, like its seed shape, the flower color, the pod color, pod shape, flower position, the plant height. It has many characters, different characters. Different 7 characters actually. These 7 characters can be studied independently of each other. It means individually Mendel could study all the 7 characters, how they are inherited. So they have many characters to follow the inheritance. The second reason was that it is an annual plant. Now since it is an annual plant, in single growing season it can be grown 2 to 3 times. It means in 1 year it can be grown 2 to 3 times. And yeah, third reason is that it has a very short lifespan. So it has a very short lifespan, so Mendel could study the characters in a very short period of time. He did not have to wait for the pea plant to grow and then he would experiment on it and study. So it has short lifespan, so he selected garden pea. The fourth reason was that a large number of seeds, it can be produced by a single plant, large seeds from a single plant. Okay, and the fifth reason, it was one of the very important reasons, that the hybrids of this pea plant, they are fertile. Usually the hybrids of any animal or plant, they are sterile. It means they cannot further reproduce, but these hybrids, they can further reproduce. It means they are fertile. So he selected garden pea. And the sixth reason was that this pea plant, it can be grown in a small area. It does not need much area for growing. It can be grown in small area. And seventh reason, you can write that this is also one of the most important reasons, that the pea plant is naturally self-pollinating because it is bisexual. It means the anther goes to the stigma and then the plants, they are crossed and then they reproduce. This is normal self-pollination. But this pea plant, it can also be cross-pollinated by hand. Cross-pollination means the pollination in which 2 individuals having different genotypes are crossed by hand. Self-pollination means the pollination in which 2 individuals having the same genotype, they are crossed. These were the reasons why Mendel selected the garden pea for his experiment. But what was the experiment? What were the steps of the experiment? Now we'll see that. What were the steps involved in the experiment? So Mendel, he conducted his experiment in 3 steps. His first step was the selection of true breeding varieties. Then step 2, he hybridized of true breeding varieties, and the hybridization of the true breeding varieties. And his third and the last step was the self-pollination of the F1 plants. F1 plants mean the first Philia Generation plants which were obtained here. Now I'll explain all this. First of all, we'll go for this step 1, which is the selection of the true breeding varieties. So for his experiment, Mendel first of all selected the true breeding varieties. Now what is meant by true breeding varieties? Now these true breeding varieties, they are the varieties that give rise to the same form of trait for a number of successive generations. It means that if these varieties they are crossed, then the same generation, it means the same trait will be formed in the further generation. If they contain the trait for tall, then their offspring will also contain the trait for tall. It means every time it is crossed, it will give the same result. The offspring, they will be same as the parents, and it will be homozygous for each of the traits. Homozygous means having the same trait, TT or TT, it means this tallness or dwarfness. So when they are crossed in the true breeding varieties, when they are crossed, then their offsprings will also be tall and tall. So in this way, Mendel selected the true breeding varieties by experimenting and self-pollinating the true breeding varieties for successive generations, so that he could ensure that their offspring are the same as the parents. Now this might be quite confusing. I'll tell you in simple words. What he did was, he chose this a variety which contained both the trait as tallness, and what he did was, these are the true breeding varieties. When he allowed self-pollination, the offspring also contained the tall. So it was purely homozygous. Homozygous means same. Homozygous and same as the parent. Again he selected dwarfness one trait. So the offspring also contained dwarfness. So in this way, when he allowed the true breeding varieties to self-pollinate among each other, the offspring also contained the same result, the same trait as the parents. He chose these both as the parents, these true breeding varieties as the parents for his experiment. So this was the selection of the true breeding varieties. While writing, you can just write that the true breeding varieties are those varieties which give rise to the same form of trait for a number of successive generations, and Mendel used these varieties as the parental generations. Then we go for the second step, which is the hybridization of these true breeding varieties. Now first of all, we need to understand what is hybridization. Hybridization, it is a process in which two individuals having contrasting characters, it means different characters, or we can say alternating characters, they are crossed. So Mendel conducted his crosses between the two true breeding varieties. It means he had the true breeding varieties as tall and the true breeding variety as dwarfness, purely homozygous, same traits. Now about the homozygous traits and alleles, these words you will understand after a few minutes when I'll teach every terminology which we'll be using in this chapter. For now, just understand that homozygous means same alleles, same traits, same characters. So it had purely tall, it had purely dwarf. So what Mendel did was, he conducted the crosses between these two true breeding varieties. And how he did this? What he did was, suppose this is the male gamete, the tallness. So Mendel, he opened the buds of the plant. Suppose this is the bud of the plant. He opened the buds in which the anthers are present. Before the anthers were fully developed, what he did, he removed these anthers from the buds and he dusted on the stigma. Stigma means the female sex organ of the plant. So he removed this anther, the male gametes, and he spread on the stigma of different flowers. And remember one term, the process of removal of the male sex organ from the immature bisexual flower, it is known as emasculation. This question may be asked in 1 mark. So what is emasculation? So it is the process of removal of the male sex organs from the immature flower. So he dusted it on the stigma, and in this way the stigma contained the dwarfness of, this is of another plant, this is of another plant, they have different contrasting characters. So he dusted the anthers on the stigma, and in this way the hybridization was done. Now the plants which was obtained as the offspring, it was planted, and then he experimented on that plant. He named it as F1 generation. It means first filial generation. So the plant which is obtained as a cross between them, it was the F1 generation, the first filial generation. When Mendel examined the first generation, it means F1 generation, he found that only one of the phenotypes was present in the generation. Now this means that all the F1 generation plants were completely tall, though they were the hybrids of tall and dwarf, TT and TT, but these F1 generation were completely tall, none of was dwarf. So the F1 generation plants, they expressed only one of the phenotypes present in the parental generation. Now Mendel carried out such 60 crosses and always obtained the same result. So this was the hybridization of the true breeding varieties. Then we have the last step, which is the self-pollination of the F1 plants. These F1 plants which were obtained. Now as I told, when two individuals having same genotype are crossed, they are known as selfing or self-pollination. Here two individuals having the same genotype were crossed. It means the F1 generation contains this trait, both tallness and dwarfness. So two of the plants, it means two of the individuals, they have the same genotype, TT and TT, it means tallness, dwarfness. So they both were crossed, and this is known as self-pollination. I'll tell you one information: self-pollination, cross-pollination. Cross-pollination is when the pollination is done by hand. It means when it is done between two individuals having different characters. But these self-pollination, it means the pollination in which the two individuals having the same genotype, they are crossed. So it is known as selfing or self-pollination. So when Mendel planted the F1 plants and he allowed it to self-pollinate, it produced a second filial generation, it means F2 generation, when these two were crossed. And what Mendel found, it was the both of the traits which was in the parental generation, they emerged in F2 generation with approximately a ratio of 3:1. Now it means that when this F1 generation were self-pollinated, then in F2 generation, both of the phenotypes, both of the traits, the tallness and dwarfness, both of the traits, they emerged in the F2 generation with exactly a ratio of 3:1. It means 3 tall and 1 dwarf. And this was the Mendel's experiment in 3 steps. Summarizingly, I'll tell you once more. First of all, Mendel did was step 1, when he found the true breeding varieties. First of all, he allowed the true breeding varieties to cross between themselves, and he assured that the true breeding varieties, they always produced the same result, the same homozygous plants. He used them as the parents, and when he used them as the parents, F1 generation was produced. And this F1 generation, though the parents were tall and dwarf, the F1 generation were completely tall. And when the F1 generation were self-pollinated, two individuals having the same genotype were crossed, then in F2 generation, again the both of the traits, they emerged, the tallness and dwarfness, both of the traits emerged. In this way we can write the Mendel's experiment in 3 steps. Now this was the step of the Mendel's experiment. Now what was the result of the Mendel's experiment? Now the first result was from this second step, that in the F1 generation, no matter how many crosses were there, how many times he conducted the experiment, only one of the characters was seen every time and never the other character. It means only the tallness, or if we use another character like round and wrinkled seeds, round is the dominant one and wrinkled is the recessive one, so every time round seeds were obtained. In this way, only the one of the generation it means one of the trait was obtained in the generation, and that one of the trait which was able to express itself, it was known as dominant trait. And another trait which was suppressed by the dominant one and it did not express itself in the first generation, it was known as the recessive one. So this was the first result. Now the second result was the trait which had been hidden in the F1 generation. It was able to reappear itself in the F2 generation, but only in a frequency of 1 of the quarter. It means this 3 1 is 4, so 1 of the quarter, 1 out of 4 times, 1 out of 4. So the trait which was able to reappear here, it was only in the ratio 1 out of 4 times. Now when we'll draw the monohybrid cross chart, we'll understand it much better. For now, just understand that the trait which was not able to express itself in the F1 generation, which was the recessive, it reappeared in the F2 generation, but only 1 out of the 3 frequencies, 1 out of the 4 frequencies. It means the total plants, if there were 4 total plants in F2 generation, then out of 4 only 1 plant could express the recessive form, not the other 3. So this is the second result. And the third result was this recessive one, it was able to express only 1 out of the 4 times. So every time the cross was done, how many times no matter, every time the ratio of dominant to the recessive, it was 3:1. It means the dominant one is always 3 out of 4 times and 1 is the recessive one. Every time we conducted the experiment, we'll understand it much better when we'll know about the monohybrid cross. So this is the 3 results. And the fourth result was, it did not matter which parent it provided the ova and which one provided the pollen. It means if this is the male and this is the female, or this is the female and this is the male, no matter whatever it is, the sex does not matters. Only matters is that the trait matters, not the sex. If this is male or female, or this is male or female, it does not matter. The first filial generation will always be the same, the second filial generation will always be the same. So the results will always same, no matter what was the sex of the parent. And this thing, it is known by a cross known as reciprocal cross. About reciprocal cross we'll be reading. For now, I'm not gonna confuse you by telling about reciprocal cross. For now, just understand, just known by the help of reciprocal cross. And this was the results of the Mendel's experiment. So with this we end our Mendel's experiment. And basically this process, the steps, they're not usually asked in examination. Still it might be asked, so I taught you. Now we're going for the terminologies and the symbols we're going to use in this chapter, which is very important for you to understand. Only then you can understand the other topics, and even this you will understand it better. So first of all, we have the term known as gene. So very often we hear the term gene, very often we use the term gene, but what is actually a gene? So gene, it is the shortest segment of a DNA. Suppose this is a DNA, okay, then the shortest segment of this DNA, it means suppose this is the shortest segment, only this much, so this is the gene. And what is the work of this gene? Gene, it is the basic unit of heredity. Each gene, it is responsible for the expression of a particular specific character. So if this is a DNA, this entire DNA contains many genes, and each gene, it is responsible for a expression of a specific character. Suppose this gene, it can express a character of either tallness or dwarfness, and suppose here is another gene, so this gene, it will express the character for anything like wrinkled seed or rounded seed, anything. There are many characters like the shape, the size, the color, anything. So all the genes, they express particular characters, specific characters. So it is the gene, it is the basic unit of heredity, and we can define the gene as it is the smallest segment of DNA that determines the biological character of an organism. Now basically DNA, it is present on a chromosome. So we know chromosome, it can be in any type. It can be acentric, it can be metacentric, it can be submetacentric. Anyway, that is not our concern right now. I'm just telling any of the chromosomes, on any of the chromosomes, the DNA is present. Suppose this is a chromosome. Suppose this is the chromosome. So this whole chromosome, one arm of this chromosome, this whole arm, it is formed of DNA, which is covered by histone protein. So this, all these arms of the chromosome, they are formed of DNA. So basically DNA is present on chromosome and gene is present on DNA. So ultimately gene is present on a chromosome, because gene is present on DNA and DNA is present on chromosome. So we can say gene is present on the chromosomes. So we say that gene is located on the chromosomes, situated on the chromosomes. So this is about gene. Now there is another term which we call genome. So what is a genome? We often hear this term genome. So genome, it is the total genetic content of an organism, the sum of all the genes present on the chromosomes of, it is known as genome, the total genetic content. So this is the genome. Then we have the term allele, and allele is one of the most important terms we're going to use in the entire chapter. We can even call it allelomorph. The actual name is allelomorph. For short form we call it allele. So what is an allele? Now allele, it is also a gene, but remember allele is a pair of alternating genes. Suppose this is a chromosome. Let's consider a chromosome, acentric chromosome. It means it does not have a centromere. Anyway, leave it. This is a chromosome. It contains a gene here. This is a gene. This chromosome has a DNA, and on the DNA there is a gene. Now this is another chromosome, it has a gene as well. So these both genes, they are alternating genes. For example, this gene controls the factor for tallness, capital T, and this gene contains the factor for dwarfness, small t. So these genes, the pair of alternating genes, they are known as allele. Now remember, allele always lie on the same locus or the same position. It means if the gene for tallness is here, then the alternating of the tallness is dwarfness, then it will lie on the same location of the another homologous chromosome. Now what is homologous chromosome? Homologous chromosome means the chromosome which is the paternal and maternal. It means paternal and maternal chromosome together is known as homologous chromosomes. The father and mother chromosome, paternal and maternal, they are together known as homologous chromosomes. So if these both are homologous chromosomes, then they have a gene for tallness here. This is the tallness. So dwarfness gene will also lie on the same locus or the same position, small t. So together we call it allele. So how allele can be defined? Allele can be defined as it is the pair of alternative genes, tallness and dwarfness, alternative. So it is a pair of alternative genes that are occupying the same locus or the same position on the homologous chromosomes. These both are the homologous chromosomes. So this is known as allelomorph or allele. So usually they have alternative genes, but sometimes in rare cases even the same gene is present. It means in homozygous condition it is present. If this is T, then it means tallness. This will also be tallness, but it is not in tallness. It is usually present in rare cases such as in parents it is present. Now that is not our concern. Just remember that if the same genes are present, the same genes are present on both the homologous chromosomes, it means the allele, it is known as the homozygous condition. Usually in parents we find this, like in the true breeding varieties we find this, like we use these true breeding varieties here, capital T and capital T. So this was basically a pair of homozygous allele, homozygous chromosomes. So with this is our fifth term, homozygous. So what is homozygous? When a diploid individual, diploid means having two chromosomes, homologous chromosomes. So a diploid individual which is carrying similar alleles, it is known as homozygous condition. Usually it is a pure form of trait. In the true breeding varieties it is found. It can be even small t, dwarfness it can be. So when two similar alleles are present, it is homozygous, and when two different alleles are present, it is heterozygous. Now you understood the term homozygous and heterozygous. Homozygous, same alleles; heterozygous, different alleles. And there is a third one which is known as hemizygous. Now hemizygous we will not be using in this chapter particularly, but there is one term called hemizygous. What we understand by hemizygous, it is that when a genotype it is determined by a single gene, These both together they determine the genetic makeup or phenotype of the organism. Like if both tall are there, then the organism will be completely tall. If both dwarf, then the organism will be dwarf. If heterozygous is there, tall and tall, then dominant one is tall, so the organism will be tall. So this is all normal one. But in hemizygous condition, what happens is that suppose individual, it has a gene. Now basically it is present in color blindness and hemophilia, so I'm going to use the gene of that. For example, this is the gene for eye. So what happens is that in color blindness, people with color blindness, there only one gene determines the character. It means out of the two genes, only one gene determines what will be the character of the organism. So these are the genes of color blindness. Like only one gene is determining, another gene is not working. So this, suppose this X gene is of the black and white, it means the rod cells. These are the genes of the rod cells, and these are the genes of the cone cells. In I, rod cells they determine in dimness, in dark light, and Y gene, it means these cells, the cone cells, they determine the color, what color the eye will see. So when this Y gene is not working, only one gene, this X gene, the rod cells is working, then the color blindness can occur. It means the people cannot identify the color, which is known as color blindness. So in hemizygous conditions, such gene, only one gene occurs. So while writing, we can just write if it is asked. Usually it is not asked. Still in 1 marks question it can be asked. So when a genotype, it is determined by a single gene, it is known as hemizygous condition. Now we have the seventh term, dominant allele. As I said, in heterozygous condition we have two different alleles. Suppose this is for capital T, tallness, and this is for small t, dwarfness. So out of these two alleles, only one of it is able to express itself in the first generation, and other one is suppressed or it is hidden by the first one, the dominant one. So this allele, it is known as dominant allele. The allele which is able to express itself, whether it is present in homozygous or heterozygous condition. Even in homozygous condition, these two alleles, they are the dominant allele because both of them are able to express themselves. So basically the allele which is able to express itself, it is known as the dominant allele. Now you must have understood this one is known as the recessive allele. So definition pretty simple. Out of the two alleles, the one which is being suppressed by its alternative allele or the dominant allele, it is known as recessive allele. So recessive allele, it is an allele which does not express itself when present with the dominant allele. Now remember, this is only in the case of heterozygous condition. In homozygous, the small t both are present, it means the dwarfness both are present, so it can express itself. This is not recessive condition. Recessive condition is only when it is present in heterozygous condition along with the dominant allele, so that the dominant allele suppresses it and it cannot express itself. Simply the allele which does not express itself is known as the recessive allele. Then we have a term, the genotype. So genotype, it is the genetic expression of an organism. Suppose we draw the monohybrid chart, we'll draw it further. So we'll see that we obtain the in F2 generation, we obtain the plants to be capital tall, it means completely tall, capital T. Then we find the dwarfness, we find the hybrids there. Now all these things, they are the genotypes. There is one pure tall, one pure dwarf, two are hybrids. So in this way, the one pure tall, two are hybrids. So in this way the ratio becomes 1:2:1, 1:2:1. So this is the genotypic ratio. Now this is known as the genotype. It means the genetic constitution of an organism. It is the genotype. We are determining on the basis of genetic constitution. But when we determine on the basis of the physical appearance, if you see the physical appearance, this is tall, okay, pure tall, one pure tall, this is also one pure dwarf. But now see, these both things, the hybrids, they are also tall because the dominant one is able to express itself, the capital T will express itself. So basically the tallness ratio will be one tall, 2 more tall, so 3 tall and 1 dwarf. So 3:1 is the phenotypic ratio. In this way determine, we'll use it in monohybrid cross and you'll understand it better. For now, just understand the physical appearance of an organism. It is known as the phenotypic ratio, or we can also call it trait. Now basically trait and phenotype are the same things. We are using the term trait. So trait means the phenotypic ratio, not ratio, actually phenotype. The phenotype means the physical or observable expression of an organism which we can see from outside. But this genotype, it is the genetic constitution, so it is not an observable phenomenon. So even if you didn't get this genotype and phenotype, just wait till the monohybrid cross and then you'll understand it completely. Now we have another term known as hybrid. We know what is a hybrid. It is the product of cross between two dissimilar parents. It is the cross between two dissimilar parents which are having contrasting characters. It means if this is the one type of parent and this is one type of parent, they are the two different types of parent, dissimilar parents having contrasting characters. One is tall, one is dwarf. Then the product obtained is known as hybrid. So it is the product of cross in between the two dissimilar parents having contrasting characters. Then we have two terms, dihybrid cross and monohybrid cross. Okay, right now I'm just telling you the definition. We'll learn about it in detail. It is one of the most asked questions. So what is monohybrid cross? First of all, monohybrid cross is a cross in which two parents which are differing in only one pair of alternating character, it is crossed. It is known as monohybrid cross. It means two parents which are differing only in one type of contrasting character. Like suppose they are differing only in the character of height of the plant. It means one has tallness and one has dwarfness. So only one type of difference is there in between the parents, and when we cross these both, this cross is known as monohybrid cross. But then dihybrid cross is another type in which two parents which are differing in two pairs of alternating characters. For example, usually we take round and yellow seeds. For example, as two characters, the shape of the seed which is round or wrinkled, and then we have the color, which is the yellow is the dominant one and green is the recessive one. We use the small letter for the recessive one. So these are the dominant, these are the recessive. So see, these both are differing in two different characters. It has a character of round, it has a character of yellow, it has a character of wrinkled, and it is a character of green. Basically they are present in pair, so let's make it in pair. So they are differing in two pairs of characters. So this type of cross is known as dihybrid cross, in which two parents which are differing in two pairs of characters, two pairs of alternating characters, it is known as dihybrid cross. So example of this will be tall versus dwarf, monohybrid tall versus dwarf. An example of dihybrid will be round yellow versus wrinkled green. Then we have a term called Philia Generation. What is the Philia Generation? We use the term F1, F2 generation. What is that Philia Generation? Actually F1, F2. Philia Generations means, see, the progeny which is obtained, progeny means the offspring. So progeny which is obtained as a result of crossing between the two parents, it is a hybrid type, and this is known as the Philia Generation. First Philia Generation. Now the progeny which is obtained by self-fertilization among the F1 plants, the first Philia Generation, they are the second Philia Generation. Simply, it is the progeny which is obtained as a result of crossing between two parents, and then F1, the hybrids obtained, they are the first Philia Generation. Then the hybrids, they are self-fertilized, and F2 generation is obtained, which is the second Philia Generation. That is, there is nothing much to tell about this. And then important term, we have Punnett Square. Now what is a Punnett Square? Basically Punnett Square, it is one of the very easy and one of the very important topics. See, Punnett Square, it is a type of checkerboard, or we can call it graphical method, in which we can see all the possible results of various crosses. I'll tell you graphically. Suppose, see, we have a cross between the pure tall and pure dwarf. These two are the crosses. We have to cross between these two, so we can know all the possible results of this combination. How we can know all the possible results? See, first of all, what we do is that male, suppose this is the male gamete, we keep the male gamete here and we keep the female gamete down. So female gamete is here. Now make a box just make a box between them. Okay, now see this T and T, so T, capital T and small t, this capital T and small t, this capital T and small t. So these are the all the results which will obtain. So this graphical representation in which we can see all the possible results of the various crosses, it is known as Punnett Square. It is very easy, so we can ensure that our cross, it is correct or not. We'll know it in monohybrid cross and dihybrid cross. See, or you can also do cross between the hybrids and see what will be the result. So if this is male, this is female, keep the male here on the top horizontal side, and on the left hand vertical side keep the female one. So now just make a box. So make a box and see what will, this is not necessary. So make a box, capital T and capital T, capital T and small t, capital T and small t, and small t, small t. So see, one is pure tall, one is pure dwarf, and two are hybrids. So this is basically this is basic information about the Punnett square. Now till now we learned about the basic terms. Now we'll learn about three types of crosses. Now these are not the three types of crosses, basically these are one among the types of crosses. Like we read about the monohybrid, dihybrid cross. Similarly there are more back cross, test cross, reciprocal cross. We'll just learn slight information about it. We are not going to use it in the entire chapter right now, at least not in this chapter. So first of all we have the back cross. Now back cross, it is a cross which involves the crossing of the F1 hybrid. First of all we obtain the F1 hybrid by crossing the two parents. So it is a cross in which this F1 hybrid, it is again crossed with one of its parents, either parent, male parent, or either female parent, parent 1, parent 2, anything. When the F1 hybrid is crossed with one of its parents, then it is known as back cross. Now what is the use of this back cross? It is a quick way to make desirable homozygous individuals. Now see how it is. Suppose this is the hybrid obtained and we are crossing it with the dominant parent, capital tall. Okay, so it will give the alleles, these, it will means gametes. These alleles will give these gametes. Now these alleles will give these gametes. So basically we'll draw a Punnett square to see all the possible results. So these both and these both. So just draw a checkerboard between them. So see, capital T, capital T, capital T, capital T, capital T and small t, capital T and small t. Now see, we got two of the homozygous conditions. Now these two of the homozygous conditions can be easily obtained when we do back cross. More amount of homozygous parents, homozygous individuals, they are obtained when we do back cross. The hybrids with the parents. We can even do it with the recessive parents. When we do it with recessive parents, it will be somewhere like this. See, T and T, T and T, but again small t, small t, small t, small t. Now all two of the dwarf parents are obtained. So in this way the homozygous condition is obtained. So this is the back cross. Usually the use is not asked, just the definition might be asked, what is back cross. So now another we have the test cross. Now test cross, it is basically a type of back cross, but it is one of the back crosses. You'll understand this. In this cross what happens is this, the individual, it means the F1 hybrid. Now in test cross we do not know the exact genotype of the hybrid. It might be capital T and small t. It might be the capital T, it might be small t, let us not say F1 hybrid. Just remember, in this cross, an unknown individual, it is crossed with one of its parents, but one of its parents is the homozygous recessive parents. So see, the unknown genotype, it is crossed with one of its homozygous recessive parents. It means it is crossed with its parents, but the recessive one parent. Now what is meant by recessive parent? The recessive parent is this one, the dominant parent is this, the recessive parent is this. So it is crossed with one of its recessive parents, and this cross is made in order to determine the genotype of this individual. Now see how the genotype will be known. Suppose we do not know the genotype of the organism. suppose the genotype is this. We do not know the genotype, okay? Suppose the genotype is completely dominant and we cross it with the recessive parents. So while drawing the Punnett square, we can see, now you can draw it on any side, it does not matter. You can draw the TT here. Now see, when we see the Punnett square, we find that capital T, small t, capital T, small t, capital T, small t, capital T, small t. So in this way, all are the hybrid dominant, all are dominant because capital T is present in all. So all are dominant. Now there is a, after doing this experiment, we find that if all the dominant results are coming, we see the phenotype. When we see the phenotype of the plant, we see all the plants are dominant, all the plants are tall. Now all the plants are tall, it means all are dominant. So if all are dominant, we come to know that the unknown generation is completely dominant. It means it is homozygous dominant. Homozygous dominant means capital tall and same capital T. So we come to know that it is homozygous dominant. Now how we come to know? Just remember, if we, now it is not a thing to remember. I mean, this is not asked what is the use of this or how we can know. Just I'm telling for your information. Only the definition might be asked for your information. See, if all the dominant results are coming, we come to know that the unknown genotype is the homozygous dominant. Now in another case, if the unknown is this one, capital T, small t, it means heterozygous dominant, and the recessive parent is this. When we draw the Punnett square, we find the T and T will come. So 50% dominant, capital T, 50% recessive, small t. So if 50% dominant result is coming and 50% recessive result is coming, we come to know that the unknown genotype, it is the heterozygous dominant, capital T, small t, heterozygous dominant. So in this way we come to know that if all the results are completely dominant, all dominant, then the individual is homozygous dominant. If the result is 50% dominant, 50% recessive, then the individual is heterozygous dominant. Now this is not of our use, you can get confused, so it is your risk, you can skip this part as well. Just remember the definition, that it is a cross in which the individual having an unknown genotype is crossed with an homozygous recessive parent. Now the last one, we have a reciprocal cross. Now the definition of the reciprocal cross is quite confusing. It is that a cross of the same genotype, it means the genotype is not changed in this cross, but the parents are reversed from the parental generations, reversed. Now see, I'll tell you what is this. It is a cross in which two crosses are done. It means, suppose parent A is there, parent B is there. Parent A is male parent and parent B is female parent. It is a cross in which two experiments are done. Remember, two experiments are done in such a way that in the first experiment parent A is used as male and parent B as female. Then in the second experiment parent A will be used as female and the B will be used as male. It means the genotype will remain the same. Suppose parent A has completely pure genotypes, capital T, and this female has, suppose, dwarf, small t, then the genotype is not changed, genotype remains the same. Just the thing is that the A will be used as male in first and in female in second, and B will be used as female in first and male in second. So it is just a experiment which is showing that the sex of the parents does not matter. What matters is the genetic constitution of the parents. So I told you about this even in the Mendel's experiment, that Mendel conducted the reciprocal cross to know whether the sex of the parents matter or not. So this is the reciprocal cross in which the first it is used as male and then female. The genetic makeup is not changed, so only male and female is changed, the sex of the parents is changed. So the definition becomes somehow like this: a cross of same genotype in which the parents are reversed. So in this way we come to know the purpose of this reciprocal cross is to find whether both the parents are making equal contribution or not. It means the sex of the parents matter or not. Yes, there are some of the exceptional cases like cytoplasmic inheritance, like sex-linked inheritance. But the sex-linked inheritance we'll be studying in the next video. For now, just understand that reciprocal cross, it is done to know whether the parents' sex matter or not. And with this we complete all our symbols and the terminologies, and these are the important terms you need to remember. Some of these terms are not needed to remember, just it might be asked in 1 marks question. And yeah, the question which is repeatedly asked in this difference between the genotype and phenotype. So I'll tell you about the difference between genotype and phenotype, and then we'll go for the 8 marks important questions, which is the Mendel's laws, and then monohybrid cross, dihybrid cross. So this is one of the important questions. Though it is not said to ask the difference between genotype and phenotype, it can be said write short note on genotype and phenotype, and then this difference will be very useful. So see, first of all, we have to write what is a genotype and phenotype. Genotype, we know it is the genetic makeup or genetic constitution of an organism, and phenotype, it is the morphological, it means outer appearance, morphological appearance of an organism. This is the first difference, basic difference. And then in the second difference we have, by knowing the genotype, the phenotype can be known, it can be decided. How? As I told you, if we have the genotypes capital T small t, then we have the genotypes the small t, and then we have the hybrids. So see, we these are the genotypes. We are seeing the genotypes. So by seeing the genotype, we can know what kind of individual it will be. This will be tall. Phenotype we came to know. This will be short. Again we came to know the phenotype. This will be again tall, and this will be tall. So in this way, by seeing the genotype, we can know the phenotype of the organism. So by knowing the by knowing the genotype, the phenotype can be decided. But by knowing the phenotype, genotype may or may not be decided. Actually, may or may not be decided. Now see how, if we are knowing the phenotype, it is tall and it is dwarf. Tall can be either pure tall, it means homozygous tall, or it can be a hybrid tall, heterozygous tall. Both are tall. Dwarf, we know that dwarf we can know. Dwarf will only be the case in which both alleles are small t. It means it will be the case only when the homozygous recessive will be there. So in this way, in this case, we know the genotype by seeing the phenotype. But in this case, we are not sure about the genotype by seeing the phenotype. So by knowing the phenotype, genotype may or may not be decided. Now genotype, it cannot be studied by direct observance. We cannot see the direct observance of the genotype of an organism. Like, what is the genotype? So we can know whether the organism is tall or dwarf by seeing it from outside, but we do not know whether it is pure tall or pure dwarf or hybrid. So it cannot be studied by direct observance. But yeah, it can be studied directly by observance. If we see the organism, either it is tall or dwarf, so phenotype can be known. And usually it comes for 3 marks, so 3 differences are enough. But one more I'll tell you. The individuals having the dissimilar genotypes, usually they have the same effect. It means two individuals, if they are having dissimilar genotypes, they may show the same effects. Like suppose this is pure homozygous tall and this is hybrid tall. So genotypes are different, but the effect is same, tall and tall. Both are showing the same effect. So two individuals having dissimilar genotypes, they may show the same effect. But two individuals having two phenotypes have different genotypes. It means that two phenotypes usually have different genotypes. Here, dissimilar genotypes may have same phenotype, same effect. Hybrid or homozygous, both have the same phenotype here. But two phenotype, they always have the different genotype. Like if one is tall and one is dwarf, so these are the two phenotypes, they have the different genotype. One is homozygous and hybrid, one is homozygous recessive, one is homozygous dominant, one is homozygous recessive. So two phenotypes, tall and dwarf are the phenotypes, they have the different genotypes. These are the basic differences between genotype and phenotype. And after this, we have the most important questions of this chapter. They are the monohybrid cross, dihybrid cross, and the laws of Mendel's. So first of all, we are going for the monohybrid cross. So now we're going to learn about the monohybrid cross of Mendel. First of all, as I said in the terminology, monohybrid cross means the cross between two pure organisms which is differing only in one pair of the contrasting characters. So first of all, when we are told to write about the monohybrid cross, we have to write the definition. First of all, it is the cross between two pure or homozygous. You can write anything. Homozygous means both the alleles are same. Homozygous, parents which are differing in only one pair of contrasting character. In place of contrasting character, you can also use the word alternating characters, because one will be pure tall and another will be pure dwarf. Or if we use other characters like color and all, then pure yellow, pure green. So this was the definition of the monohybrid cross. But here the cross is made in a single contrasting character, which is the plant height. We'll be dealing about the plant height. So it means tall pea plant and dwarf pea plant. Now a tall and a dwarf pea plant are selected. Tall pea plants, they are designated by capital letter T's, 2T, and they are designated by small letter t. Now it is an assumption that the factors or the genes, they exist in pairs. It is just an assumption, because in diploid organism they must exist as pair, it is said so. So what happens is that meiosis takes place in both of them, and then gametogenesis, during the gametogenesis meiosis takes place, and they both separate and form single gametes. This is the meiosis division during gametogenesis. It means the gametes are formed. See, these are the parental stalks which contain the alleles capital T and small t. So when gametes are formed from one parent, two gametes are formed. So the alleles, they go respectively in each of the gamete. Like one allele goes to one gamete and another allele goes to another gamete. Similarly in dwarf. Now when these gametes are fertilized, then a zygote is formed with a phenotype capital T and small t. This is the hybrid formed. When they all fertilize, these type of zygotes are formed. Now we have to represent it in the figure. First of all, we have to write these details in small paragraphs. Even this detail we can write in paragraph, like the tall pea plants, they are designated by capital letter T and dwarf by small letter t. So during gametogenesis meiosis takes place and two gametes are formed. When these gametes are fertilized, the zygote with the genotype capital T and small t, this hybrid is formed, which develops into a seed. Now in the figure we can see, first of all, we have the parent pure tall. This is the pure tall, and this is the pure dwarf. And trust me, monohybrid cross and dihybrid cross are absolutely easy, nothing much harder. The harder part was behind. These are just easy things. Now meiosis takes place and forms the gametes. So these are the gametes. So we'll keep on writing on the left hand side what are these things. So this is the phenotype, isn't it? Tall and dwarf. So phenotype of parents. Now this is the genotype of parents. Now what happens? This meiosis division took place here, and then these are the gametes. So these are the gametes. Now fertilization takes place. Now while showing the fertilization, don't be confused. We have to show. I'll show you how we have to draw the chart. First of all, join these both, okay, and then join these both. So join this with this, and again join this with this. Now similar will be with this. First of all, join it with this one, and then join it with the last one. In this way we are joining both of them with both of them. So in this way, T, capital T and small t, one capital T small t, again this capital T and small t, again this capital T and small t, again this. So we see that this is the fertilization process, the random fertilization. Now we'll read about this random fertilization in the Mendel's laws also. The last topic will be the Mendel's law of inheritances. Now those Mendel's law, they are just dealing about the monohybrid and dihybrid cross. When we are set to write about the Mendel's law, we have to write about the monohybrid cross and dihybrid cross. So I'll tell you about that later on. First of all, let's focus here. This is the fertilization took place, and this is the genotype of the zygote formed. Genotype of zygote. Now if we see, this is the tall, because capital T is dominant one, so tall, 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 tall. All these are hybrids, but the dominant one is capital T, so all are tall. This is the phenotype of the zygotes. So in this way we see that all the F1 generation, first filial generation, they are just tall. These all are the F1 generation. We see they all are the hybrid tall. So now further, first we drew this much, we wrote about this much. Now we have to write about the second process. Now what happens is that when these F1 tall, hybrid tall generations, they are allowed for self-pollination. Now again I'm clearing self-pollination and cross-pollination here, so you do not get confused. Self-pollination is when two individuals having the same genotype are crossed. See here, the capital T and small t is present, even here capital T and small t are present, so they both have the same genotype. So when they are crossed, it is known as self-pollination. But when these are crossing, they have different genotype. This is known as cross-pollination. When we do with the help of our hand, when we do artificially, it is the cross-pollination, and this is natural. The self-pollination occurs naturally, the same genotype. So when the two hybrid tall generations, they are the two hybrid tall of F1 generation, they are allowed for self-pollination. The plants produced, they are the F2 generation. So let's go for that. Now this is over. Then two hybrid talls are taken. This is hybrid tall. This is hybrid tall. So again, this is the phenotype. This is the genotype. Now again, gametes are formed. Here meiosis took place, and then gametes are formed. Gametes formed are different this time. Now fertilization will take place. The process of fertilization will be similar. First we have to join with this, and then with this, then with this, and finally with this. So this time when we see, this is the fertilization, and here the zygotes are formed. So when we see the zygote, capital T and capital T is formed. Then we see capital T and small t is formed. Again capital T and small t is formed, and finally small t is formed. So this time the zygotes formed are different. This is the genotype of the zygote, and when we write the phenotype, this will be tall. Even this will be tall, because since it is hybrid, the dominant one is capital T. Again this will be tall, but this will be dwarf. So this time when we see the phenotype, it is different from the F1 generation. This time the phenotype has one dwarf and three tall. So the genotypic ratio here, first of all you'll see the phenotypic ratio. See, tall, tall, tall and dwarf. So our phenotypic ratio, it will be 3 tall and 1 dwarf. So 3:1 is our phenotypic ratio, 3 tall and 1 dwarf. But the genotypic ratio, pure tall, hybrid tall, pure dwarf. So our genotypic ratio, it is 1 pure tall, 1:2 hybrid talls, 2:1 pure dwarf. So this is the difference between phenotypic ratio and genotypic ratio. The phenotypic ratio here is 3:1, 3 tall and 1 dwarf. The genotypic ratio 1:2, 2:1. 1 homozygous pure tall, pure means homozygous. So homozygous pure tall, 2 dwarf tall, and 1 is pure dwarf. We can even call it heterozygous. So these are the F2 generations which are formed by the self-pollination of the two hybrid tall of F1 generation. So this is about the monohybrid cross. While writing, we just have to explain this thing, and explaining this thing is not a big deal, it's very simple. So finally we just have to write the conclusion about the monohybrid cross, which was drawn by Mendel. And yes, again I'm telling you, the description of this chart is not much difficult. Just writing that one pure tall and one pure dwarf is taken, gametogenesis takes place, it means meiosis, division takes place, and two gametes are formed. And when the gametes are randomly fertilized, F1 generation is formed, which is completely hybrid tall. When the two hybrid tall of F1 generation, they are allowed for self-pollination, then F2 generation is formed. In F2 generation, we find the phenotypic ratio to be 3:1 and genotypic ratio to be 1:2:1. So this is about the monohybrid cross, and then we have the conclusions drawn by the monohybrid cross. So the first conclusion which is drawn by Mendel from the monohybrid cross, it was that the F1 plants, they displayed only one of the phenotype from the parents. They displayed only one phenotype of only one type of parent, either tall or dwarf. Here we are seeing it is displaying only tall. In the F1 generation, we saw it is displaying only tall. So it is displaying the phenotype of only one of the parent. It displays phenotype of only one parent. But when we see in F2 generation, we see that the dwarf is also present, and we are getting dwarf from the same F1 generation. When we self-pollinate the F1 generation, we get dwarf as well. So Mendel concluded that though it is only showing phenotype of tall, only one parent, it must contain the genotype of also the dwarf one. Only then in F2 it is showing the dwarf. So the presence of both tall and dwarf seeds in the F2 generation could be explained only if the F1 generation possessed both the tall and dwarf genes. So he concluded that these plants, they contain two genetic factors for coding for a character. It means that he concluded that these plants are containing two genetic factors. On the basis of that conclusion, it is written capital T and small t, the two genetic factors are written in the zygote, because he concluded that the two genetic factors must be present in the plant. Only then they are showing even the dwarf one in the F2 generation. So this was the first conclusion. So it is not much confusing, just it is simple that, see, Mendel, he saw that the F1 generation is showing only one of the phenotype of the parent, but again in F2 generation the phenotype of the second parent is also seen. So it can only be explained if the F1 generation, it is also containing the genotype of the dwarf one. So he concluded that it must contain two genetic factors which are encoding for its character. So this is the first conclusion. And the second conclusion was, it was the concept of dominance. So he said that in F1 generation, the phenotype was seen only of the one parent. So the trait which disappeared, the phenotype which disappeared, it was known as the recessive one. And the phenotype which was able to show itself, the trait which was able to show itself, it was known as the dominant one. So it is the concept of dominance which is the second conclusion of Mendel. It is very simple, you can write about it. Then the third one conclusion was that the two alleles which are present in each plant, they separate at the time of gamete formation, and each allele goes to one of the gametes. This was the third conclusion. It is an important conclusion which we will be using in the Mendel's law as well. So listen here again. See, the parents contain two alleles, capital T, they are in pair. It means one allele is in pair. So the two alleles in each parent, the pure tall alleles and the pure dwarf alleles, the two alleles in each parent, they separate at the time of gamete formation, and each allele goes to one of the gametes. So see, each allele is going to one of the gametes. So this is the third important conclusion of Mendel, and this is also the law of segregation of gametes which will be using in the Mendel's principles of inheritance or Mendel's law of inheritance. It is also one of the repeatedly asked questions, but here we do not need to explain that. Just we need to write a simple line that the two alleles in each parent separate when the gametes are formed, and each allele goes to one of the gametes. In the bracket you can write law of segregation. So this will be enough for the Mendel's conclusion. Three conclusions are enough. So this is about the monohybrid cross. Now we are going for the dihybrid cross of Mendel. So dihybrid cross is a cross between two homozygous parental stalks which are differing in two pairs of the contrasting characters. So first of all, definition: the cross between two homozygous or pure, you can write anything. Homozygous is much scientific, so we're using homozygous parents differing in two pairs of the contrasting or alternating, anything you can write, contrasting or alternating characters. It is known as dihybrid cross. Now in this cross, the two contrasting characters which are considered, they are first of all the shape of the seed, and then the second one is the color of the seed. So in shape, when we see the shape of the seed, it can be either round or it can be wrinkled, and when you see the color, it can be either yellow or it can be green. So see, round, it is the dominant one, wrinkled is the recessive one. In this, yellow is the dominant one, green is the recessive one. So when we see what Mendel took for his experiment, it was the two breeding varieties which differed in this characters: double dominant round and yellow seed, and second was the double recessive, wrinkled and green. Now what does this mean? It says that Mendel took such seeds in his experiment that was the first seed was round and yellow, the second seed was wrinkled and green. So it is known as double dominant because see, round is also the dominant one and yellow is also the dominant one. So the first one was a double dominant, round and yellow. The second one was the double recessive, wrinkle is also recessive, green is also recessive. So double recessive, wrinkled and green variety of seeds. So these two were the varieties of seeds which Mendel took for his experiment. When we write in the symbolic form, so round will be denoted by capital R. We said that it is an assumption that it occurs in the pairs. So capital R, and then yellow, capital Y. So this is the double dominant, and then for recessive one we use the small letter of the dominant one. So see, wrinkled means small r, green means small y. So these are the two varieties of seeds Mendel used. Now what happens is that in this dihybrid cross, a pure round and yellow seed and a pure wrinkled and green seed, they are crossed with each other. The gametes produced by round and yellow seeds, they are represented by RY, and the gametes produced by wrinkled and green seeds are RY. I'll show you how it is. See, first of all, the two pure varieties are taken, round yellow and wrinkled green. So we can write here it is round yellow, which is the phenotype, and wrinkled green, which is again the phenotype, and this is the genotype. So now we know meiosis will take place during the gametogenesis and gametes will be formed. So gametes formed, RY goes to one gamete and RY goes to one gamete. Again, even here, small r and y, small r and y. So these are the gametes which are formed during gametogenesis. I told you about this. These are the gametes which are formed during gametogenesis. So when these both gametes, they are allowed for cross fertilization. Again, cross fertilization because see, they are different from each other. It fertilizes with this, it fertilizes with this. So different gametes are fertilizing, different genotypes are there, they are fertilizing. So it is known as cross pollination. So when they are allowed for cross pollination in F1 seeds, we see this type of structure. It will cross pollinate with this, it will with this, and then this. So when we see capital R, small r, and then capital Y, small y. Now again capital R. So almost it will be same thing everywhere. You can write it anyway. I'll write it this way. Okay. So again, this is the fertilization, and these are the gametes from the genotype of the gametes. Now when we see the genotype of the gametes, we find that these plants, again round and yellow, both the varieties are present, both the genes are present in it, but the dominant one will be able to show itself. So in round, capital R is present, so it will be round, and in yellow, capital Y is present, so round yellow. Now same thing is present in all of them, so all of them are the round yellow seeds. It means all of them are hybrid round yellow seeds. Now it is similar, it is corresponding to the hybrid tall varieties. In hybrid tall, both the genes are present, capital T and small t, but the phenotype will be tall because the dominant one will be able to show itself. So in this also, both the genes are present, but the dominant one will be able to show itself. So the genotype and the phenotype, we can see the phenotype will be all same, all round yellow, but the hybrid, so they are heterozygous. Hybrid means heterozygous, so they are double heterozygous, we call it, because it was double dominant, double recessive. Even here double heterozygous, because two things are heterozygous, the round one and the yellow one, two characters are there. So double heterozygous round yellow, we can call it, or simply hybrid. Now as we did in the monohybrid cross, we'll allow them for self pollination or self fertilization. So now they are gone for self fertilization. We'll do it on the page. So these are the F1 generations, two F1 hybrids, they are allowed for self pollination. Now you can see one hybrid, each of the plants, they produce 4 types of gametes. If you see properly, it will produce 4 types of gamete. First of all, capital R, see the combinations, there are 4 types of combination, capital R and capital Y, okay, and then we have capital R and small y, then small r capital Y, and small r small y. So these are the 4 types of gametes produced. Now don't write capital R and small r, it is not a gamete. We are doing double dominant, double recessive, double hybrid. It means we are doing 2 of the characters, so 2 of the characters must be presented, 2 different characters must be present, because it is a dihybrid cross. So when we see 2 different characters are present, it can be present in 4 possible ways. The capital R, capital Y, capital R, small y, small r, capital Y and small r, small y. The similar one will be present in this one. So we cannot do this cross now here, we cannot make it cross, it will be much complex. So for this we are using a Punnett square. Punnett square, as I told you, it is a very easy method for knowing all the possible combinations that will be produced between 2 crosses. So we'll use the Punnett square here. If we are drawing the same chart as we did in the monohybrid cross, like drawing these charts, it will be much complex. So we'll just draw Punnett square. First of all, one parent here. So if you see, one parent is kept in a horizontal row and another in the vertical row, sorry, column. This is capital Y, isn't it? I wrote it small y. So now just we have to draw a checkerboard, and we have to keep the combinations. So now see the combinations, capital R, capital Y, capital Y, and then we can see capital R, Y, Y. We are just keeping the combinations. Then you can do it along with me, so it will be easier for you to understand as well. We are just multiplying these both things. So after completing the Punnett square, you will find some structure like this. So again, I am clearing from the first one. Don't be confused with the monohybrid and a dihybrid. In monohybrid, just we did was the pure tall the gametes were produced, then the tall hybrids were produced, 2 hybrids were self fertilized, and then the 1 tall, 2 hybrid tall, and 1 dwarf was produced. But in dihybrid, we just did round yellow. So again we fertilized, then hybrid round yellow was produced. Now again similar, we are self fertilizing this hybrid round yellow, but here the 4 gametes are formed, so we cannot do as we did in the monohybrid cross, simply a chart, because the 4 gametes are produced. So for this we are drawing a Punnett square. So simply these combinations are produced. These combinations, they are corresponding to the combinations which are produced at the last in the monohybrid, the tall, the hybrid tall, the pure dwarf. So it is corresponding to this one. So these are the genotypes. When we write the phenotype, we can see this both of them are round, and these are also yellow, so they are round yellow. So round yellow is there. Similarly, this will also be round yellow. This will be round yellow as well. The dominant one is present. This will also be round yellow, and this will be round, but see here both of them are recessive one. Recessive means green, so round green. Even here round yellow, but here again round green, because no capital Y is present. In similar way, you can write all the phenotypes there. So after writing the phenotypes, we'll find that total out of these 16, we find 9 are the round yellow one. So see, round yellow, they are 9 out of 16. We'll see wrinkled green to be just 3 out of 16. After you write, you will understand it will be 3 out of 16. But now we'll find two new varieties. See, these are the wrinkled green one, but this is wrinkled yellow, capital Y is present. Similarly, this is round green. So see, two new varieties are formed, round green and wrinkled yellow. Now why it is called two new varieties? You see the parents. In the parents, round yellow is there, wrinkled green is there, but in the parents, round green and wrinkled yellow is not there. It means the parents we used were round yellow and the wrinkled green, but here two new combinations came in the offsprings, which are round green and wrinkled yellow. So these new combinations in the F2 generation, this is our F2 generation, these new combinations in the F2 generations, they are known as the recombinants. So remember the term, this is an important term, recombinants. These both are the recombinants because they both were not present in the parents, but they appeared in the offsprings. So the number of both of them will be 3 out of 16, 3 out of 16. When we write this, we'll understand. So total out of 16, round yellow is the most, 9, and rest are 3, 3 and 3. And this is not 3, sorry. Even I got confused. It is 1. I didn't write, so I got confused. It is 1. Only one wrinkled green is present, and rest all nowhere wrinkled green is present. So 9, 1 and 3, 3. So in this way, when we see the proportion, when we write it in the phenotypic ratio will be 9 is to 3 is to 3 is to 1. Because see, 9 is to 2 round green, 3 is to 2 wrinkled green, 3 is to 1 wrinkled yellow, and then 1 wrinkled green. You understood this, yeah? 9 is to 3 is to 3 is to 1. Simple phenotypic ratio. Now what will be the genotypic ratio? Genotypic ratio here is a bit complex, because see, so many genes are different types of genotypes are present here. Phenotypic ratio was simple, 9 is to 3 is to 3 is to 1. When we look at the genotype, we can understand the phenotype and then write the phenotypic ratio. But now the genotypic ratio is a bit complex because many type of genotypes are present. So first of all, we'll draw a table to write all these genotypes clearly so that we can understand what is the genotypic ratio. So see, now genotypes of the F2 generation. When we see the genotypes in the table, first we'll write about the double dominant R. So see, capital R, capital Y, capital Y. This is the first type. The second type, capital R, capital Y and small y, the second type. And then the third type, again it is capital R, small y, small y, small y, small y. And then in similar type, we are just writing all the types which are present. So when we write all the types which are present, it will be somehow like this. If this type is present, even this one type is present, even this type is present, and then small r type is also present, and smaller y. So these are the different types of genotypes which are present in this table. Now the number of genotypes which are present here, this capital R and capital Y, 1, and nowhere else it is, so number is 1. Capital R, capital Y and small y, 1, 2, yeah, only 2 are present. So in this way, when we count all of them, this is 1, 2, this is also 1 type, this is 2 type, these are present in 4 type. Capital R, capital small r, capital Y, small y. 1, 2, and then 3 and 4. Yeah, so 4 types are present, and similarly others are 1, 2. Sorry, these are 2, these are 1 and 2 and 1. So in this way all the numbers are present. So these are the, now we'll just write the genotypic ratio, we'll just write these numbers. Actually, what is genotypic ratio? Genotypic ratio is the ratio of the types of genotypes present. It means, as we wrote in the monohybrid cross, one type of pure tall, so 1, 2 types of hybrids, so 2, 1 type of dwarf, so 1. The types of genotypes which are present, the types of the genotypes which are present in the F2 generation, they are the genotypic ratio 1:2:1. So similarly here the types which are present, they are the ratio. So genotypic ratio will be, just write 1:2:1 is to 2:4:2:1:2:1. It means 1:2:1 is to 2:4:2:1:2:1. This is the genotypic ratio. Quite long, isn't it? It is very confusing type, so I told you with this table. In the examination, it's your wish. You can draw the table, you cannot draw the table, but if you draw the table, it will be easier for you to write about this. Now remember, this the order should not be altered. Now, so that you do not alter the order, I'll tell you one trick. First of all, always write about the capital R. Okay, first complete all of capital R. Like first complete where 2 capital R's are present, write about those. 2 capital R, 2 capital R, 2 capital R. We wrote about that. Just write in order from greatest to the lowest one. So then write about the capital R, small y, but then see the order is both capital Y, then 1 capital Y, small y, then both small y, small y. So in this way you can understand, you just write according to the order. So in this way we complete the dihybrid cross. So again I'm telling you from the beginning, in the dihybrid cross, when we are writing about the dihybrid cross, about the chart you understood clearly, I know. Now see, while writing about the dihybrid cross, we just write the first of all the definition, and then we write the information that Mendel took 2 varieties which are the double dominant round yellow and double dominant wrinkled green. So the gametes were formed, and then the zygote contained round and yellow, but they were hybrids. It means double heterozygous round yellow. So when they were allowed for further self-pollination, then each of the parent, they produce 4 types of gametes. So these 4 types of gametes, then the Punnett square is to be drawn, and finally we see the genotypic and the phenotypic ratio. So on the basis of that, the conclusion of dihybrid cross, it means as we did in the monohybrid cross, the conclusions, similarly here conclusions of the dihybrid cross which Mendel took out. The first conclusion was in that F2 generation. In F2 generation, 2 new combinations are formed. The 2 new combinations, round green, wrinkled yellow. So 2 new combinations are formed known as recombinants, which are different from the parents. Second one is that the phenotypic ratio is found 9:3:3:1. And the third one was that the genotypic ratio is found is to be 1:2:1, this one. So these are the conclusions of the dihybrid cross, and with this we complete our dihybrid cross. And it was pretty simple, the monohybrid and dihybrid cross, but it is one of the repeatedly asked questions. Dihybrid cross is much probable than monohybrid cross. Studied properly. And now we have the last topic, which is the most important one in this chapter, the Mendel's laws of inheritances. So we have the Mendel's law of inheritance now. On the basis of the monohybrid and the dihybrid crosses, Mendel postulated 2 laws which are now called after his name, which is the Mendel's law of inheritance. So it is on the basis of the monohybrid and the dihybrid cross which we learned earlier. So these laws, first of all, we have the first law, which is the law of segregation. Law of segregation, or it is also known as the law of purity of gametes. So now remember, these laws, they are definitely gonna asked in the examination, either in the 8 marks or either in 3 marks. It will definitely ask. So law of segregation, what it states. Now, this law states that each diploid organism, it possesses 2 alleles for any particular character. We learned it earlier, that any organism, it has 2 alleles for any particular character. It means, for example, this capital T and small t. So each organism, this is one organism, this is another 2 parents. So each organism is it is containing two alleles for any particular character. Now while the gametogenesis, during the formation of the gametes, these two alleles they segregate means separate. So these two alleles separate and one allele goes to each of the gametes. During gametogenesis, two gametes produced, and each allele it goes to one of the gametes. Now this is basically the conclusion which we learned in the monohybrid cross, and I said this law is on the basis of the monohybrid cross. So this is the law of segregation. So while writing, just simple that this law states that each diploid organism possesses two alleles for any particular character. These two alleles segregate or separate when the gametes are formed, and one allele goes to each of the gametes. So it is known as law of segregation. Now why it is known as law of purity of gametes? Now see, it is called purity of gametes on the basis of the F1 generation. See, in the F1 generation, when we see two alleles are there, but one is capital T and one is small t, it means one is the character of tall, one is the character of dwarf. So when we see the hybrid, it has two. This is the F1 generation. F1 generation, both of the characters are present, and while the formation of gametes in further self-fertilization, these two separate and form two gametes in which both of them go. So in this hybrid, we see that both the tall and the dwarf alleles, they are together present, but they are not blending or mixing with each other. They are present independent of each other. They are present purely because at the time of gamete formation, we see that pure tall goes to one and pure dwarf goes to another one. So in the F1 generation, they have two contrasting characters of dominant and recessive type, but these factors remain together without blending or mixing with each other, and this is why it is called the law of purity of gametes. Okay, so the law of segregation, just you have to write why it is called law of segregation, and then why it is called purity of gametes. So this was about the law of segregation of gametes, but further if it is asked in long question, you even have to give the example. So while giving example, you have to explain the monohybrid cross again. Now it's not much bigger deal to explain the monohybrid cross. Now you do not need to draw the figure of the monohybrid cross, just you can explain in a simple paragraph and that will be enough. Like you just have to tell what happens in the monohybrid cross, that Mendel demonstrated a hybrid between Mendel demonstrated the cross between two pure tall and pure dwarf. So gametes were produced during gametogenesis, and the pure tall alleles, one allele went to each of the gametes, as we said in this, that each allele it goes to one of the gametes. So each allele went to each of the gametes, and then further hybrids were produced. When the hybrids were further fertilized, then the F2 generation were produced, which contained even the dwarf characteristics. So in hybrid, both of them remained without mixing with each other. That's all about the law of segregation. So finally, what is the law of segregation? It states that two contrasting characters or alleles, we can say two contrasting alleles which are present in a heterozygote or hybrid individual. It means two contrasting alleles which are present in a heterozygote or hybrid individual, they do not mix up, but keep their identity distinct. It means they are keeping their identity distinct, they are not mixing up, and at the time of gamete formation, each of the allele they segregate or separate, and each allele goes to one of the gametes. So this was the complete summation of the law. Again I'll repeat, the two contrasting alleles which are present in the heterozygote individual, they do not mix up with each other but keep their identity distinct, and they separate or segregate out at the time of gamete formation, and each allele goes to each of the gamete. Okay, that was about the law of segregation. Then we have the last law, which is the law of independent assortment. So see, the law of independent assortment is a bit confusing. You can get confused, but I'll try to make you clear. See, this law states that the different characters located at the different positions of the chromosomes. So first line is a bit confusing. First we'll see. It states that the factors, the genes of different characters which are located on the different pairs of the chromosomes, they are independent of one another during the gamete formation. This line means that suppose this is the chromosome, okay, so here is one character suppose this character, it determines the shape of the seed. Suppose this is the character of round and this is the character of wrinkled. Okay, and on the second position, I told you that at a single position only one of the character is present on a chromosome. So on the different position there is a character of color of the seed. So this is yellow, this is green. Okay, so we have two characters on the chromosomes. Now remember, law of independent assortment is about dihybrid cross. Segregation about the monohybrid. Independent, the second law, it is about the dihybrid cross. So these two characters, they are independent of one another during the time of gamete formation. When we have gamete formation, the round yellow and the wrinkled green, they both characters, they are independent of each other. The shape of the seed and the color of the seed, they are independent of each other, because at the time of gamete formation, after the gametes are formed, these gametes will be formed first of all, and when we find the F1 generation, we find it will be somehow like this. So these both characters, they are independent of each other and they show independent dominant effects. So see, this round and yellow, they are independent of each other. They show independent dominant effects, because round is also dominant, yellow is also dominant. So if we see the F1 generation, it is both round and it is yellow. So it is also dominant, it is also dominant. They are showing their independent dominant effects. The shape of the seed is showing its independent dominant effect, and the color of the seed is showing its different dominant effect. So they both are independent of each other and they show their independent dominant effects. So this is the law of independent assortment. Further, if we see, we have to give the example of the independent assortment, because when it is asked in long question, the example is very necessary. Only this much information, it is not enough about the law of independent assortment. This will be merely 5 or 6 lines. So when we are giving the example of the independent assortment, we have to give the entire example of the dihybrid cross. We have to explain the entire dihybrid cross. Now the figure is not necessary, only the explanation. Again, I will show you the explanation here. So it is very simple. When a pea plant having two different contrasting characters, the round yellow and the wrinkled green, they are crossed with each other. Cross pollination is occurring. They are crossed with each other. In the F1 generation, the hybrids are produced which are showing round yellow. They are the heterozygous individuals. Now when they are fertilized, they produce 4 types of gametes. As we said in the dihybrid cross, they produce 4 types of gametes, which are the RY, capital R, capital Y, capital R, small y, and in similar way both of them produce the 4 types of gametes. And the F2 generation, which are produced by self-pollinating the F1 hybrids, we can see these are the types of phenotypes, genotypes and phenotypes produced. So then we just have to write about this thing. The round yellow are this much, the recombinants are this much in number, and the phenotypic ratio and the genotypic ratio, and that's all about the law of independent assortment. So again, I am repeating the laws of inheritance. First law, law of segregation. It states that the two contrasting alleles which are present in the heterozygous individual or hybrid individual, they are independent of each other. Okay, they are independent of each other, they do not mix with each other, and they separate at the time of gamete formation, and each allele goes to one gamete. This is the law of segregation. Give the example of monohybrid cross. Law of independent assortment. The two contrasting characters, the two different characters which are located at different pairs of the chromosomes, they are independent of each other and they show independent dominant effects. Okay, and give the example of dihybrid cross. And with this we complete our chapter. This chapter is one of the very important chapters, because as I said, every year questions from this chapter is asked, and it's sure to be asked this time again. And Mendelism, it is an important topic because Mendel's laws, they are of great use in solving problems of the genetics, like dominant and the recessive characters could be found out after this. Then the crosses could be made to produce the hybrids of the desired characters. Agriculturally useful plants, they can be improved. Pure but recessive characters can be used wherever required, and genotypes and phenotypes can be predicted. So these were the importances of the Mendelism. again, I'm summarizing the entire chapter. First of all, we read about the reasons for selection of garden pea. Then we read about the steps of Mendel's experiment. Then we went through the results of the Mendel's experiment. Then I taught you some symbols and the terminologies. And then we have the important difference between genotype and phenotype. Then we have monohybrid cross. Then we have the conclusions of the monohybrid cross. Then dihybrid cross, conclusions of the dihybrid cross. And finally we have the laws of the Mendel's experiments. So this was about the chapter. Now the next genetics video is coming soon. Till then, take care. Goodbye.